In this video, we will discuss about SARS-CoV-2 spike protein and ACE2 binding study and why it is important. So far, we have understood that SARS-CoV-2 use ACE2 receptor that is present or that are present on the human cells to get inside the cell and take care and take over the machinery of the cell to reproduce itself. However, so far, people have not understood in detail how this SARS-CoV-2 and ACE2 binding occurs. The main thing that people have understood that the non-self epitope that are present on SARS-CoV-2 spike protein are been masked by the self-glycans, as SARS-CoV-2 also have glycan structure similar to the humans. Even though 95% of this SARS-CoV-2 sequence matched with the matched with the SARS-CoV virus present in the bat. So, in order to understand the detail of SARS-CoV-2 spike protein and ACE2 binding, it is essential to understand the glycosylation pattern and the heterogeneity at specific site of the both the spike protein and the ACE2 binding, so that vaccines can be produced or the strategies can be developed in a manner that are much more effective to prevent SARS-CoV-2 virus to come in contact or come in efficient contact with the ACE2 receptors that is present on the human cells. So what people have done is that they have separated the soluble part of ACE2 with the help of ADAM proteases, and then purify the soluble part that is separated from the membrane of the plasma membrane of the human cells. And in this soluble ACE2, they have found out that it has 6 end-linked glycan sites and several O glycan sites. At the same time, they have also developed the soluble stable trimeric part of spike protein and understand its glycosylation pattern to find out how the uh, heterogeneity or glycosylation heterogeneity at different site of glycosylation are affected, or so that are affected, as this occupancy and site specific heterogeneity is important for developing a neutralizing antibodies. So in this figure, we can see we can start from the A. So this A panel of the figure shows the sequence of amino acid that are present in soluble ACE2 that has been uh, synthesized, that has been stabilized and synthesized and used further in this study. In this we can find that the red highlighted asparagine is 6, and these asparagines are in order that it aligns with the signal, it aligns with the signature sequence of S for end-linked glycan, that is NXST site, where X is not equal to proline and S is serine and T is threonine. For example, this is NIT. I is any amino acid except proline, and T is one of either T or S. And this Q is also highlighted, where the where pyroglutamine uh, uh, where pyroglutamine formation occurs, where peptide pyroglutamine formed attached to this Q site, where pyroglutamine protein attached to this Q site of the soluble ACE2. And similarly, in SARS-CoV-2 trimer we can see that, and this soluble ACE2 is after the ACE2 that has been separated from the plasma membrane of the cell after treatment of ADAM. And in this SARS-CoV-2 trimer that has been prepared, stabilized, and prepared, that has been stabilized in a manner that it resembles more close to the actual SARS-CoV-2 spike protein. In this also, the N is the highlighted uh, asparagine linked signature sequence, and there are total 22 N linked asparagine linked sites. And this Q is also uh, in this Q or glutamine represent the amino acid where uh, pyroglutamine protein attached, and and that has been used uh, as a strategy to further obtains the proteome further obtains the different LCMS analysis such as glycomics, proteomics, glycoproteomics. And this is the electron spray microscope. uh, uh, this is the negative electron microscope, image of the SARS-CoV-2 trimer, just to confirm that our trimer resembles with the actual trimer. They have also used the Western blot with the help of actual anti-spike, uh, antibodies, anti-SARS-CoV-2 spike antibody, and SARS-CoV, and at the same time, Western blot is also done for anti-ACE2 antibodies. And just to confirm that whatever the ACE2 and SARS-CoV-2 trimer used in the experiment is uh, resembles to the actual ACE2 and SARS-CoV-2 spike protein. And this panel D shows the proteomics and glycomics analysis. Here we can see that the blue and red represent the peptides that contains represent the peptide that contains the asparagine linked sequence and glycan sequence, and this pink or the purple color shows the glycan, the purple color in the MS1 spectrum shows or purple color in the spectrum shows the glycan data present in the sample, and this green color shows the peptide that has the either has partial or full glycan sequence present. So this figure shows the authenticity of the SARS-CoV-2 spike and ACE2 that are being used in this study, and some of the characterization of the glycomics profile of the spike protein with the help of pyroglutamine, with the help of pyroglutamine protein, and after uh, trypsinization of this pyroglutamine protein, this analysis has been done, this proteomics and glycomics analysis. So that's all for this video. Thank you for your time.